Hello guys, welcome to my channel. Myself Vaishnavi, and today's session now we are going to see the topic about Enterococcus faecium. First we will discuss about generally about the Enterococcus faecium and where it is present. Enterococcus faecium is a bacterium. It is commonly found in the intestines of humans and also the animals. While it is usually harmless in the healthy individuals, it can pose threat to individuals with or weakened immune system. Enterococcus faecium is known for the resilience and ability to survive in various environments, including hospital, where it can sometimes cause infections that are challenging to treat due to the antibiotic resistance. And the morphology of Enterococcus faecium. Enterococcus faecium is a gram-positive bacterium with a spherical or ovoid shape. It typically occurs in pairs or short chains. The cell wall of Enterococcus faecium contains peptidoglycan, giving it is a rigid structure. Additionally, it has a capsule that surrounds the cell wall, providing the protection against host defense. The bacterium is non-motile and does not form spores. And then cell shape and arrangement. Enterococcus faecium is typically appears as a cocci. When observed under a microscope, they often occur in pairs or short chains, exhibiting a typical cocci arrangement. And then discuss about the size of Enterococcus faecium. The Enterococcus is generally 0.5 to 1.5 micrometer in diameter. And then capsule. Capsule is not present in the Enterococcus faecium. And then flagella and motility. Enterococcus faecium is generally considered as a non-motile, which means it lacks flagella, it does not move. Growth characteristics of Enterococcus faecium. The temperature range of Enterococcus faecium is 10 to 45 degrees Celsius and the pH is 9.6 to 5. With an optimum pH is 7.4. And the oxygen requirement of the Enterococcus faecium is facultative anaerobe. And then salt tolerance. It exhibits tolerance to higher salt concentration and allow it to survive in environments with elevated salt levels. And then Enterococcus faecium is known for the ability to form biofilms, and this biofilms formation can contribute to its precedence in various environments. And Enterococcus faecium can utilize a variety of nutrients for growth, including sugars and also the carbohydrates. Staining of Enterococcus faecium. Enterococcus faecium is a gram-positive, which means it retains the crystal violet stain in the gram staining procedure. Pathogenesis of Enterococcus faecium. Enterococcus is a bacterium that can cause infections in hospital settings. Its pathogenesis involved factors such as antibiotic resistance and biofilm formation, the ability to adhere and host tissue. Enterococcus faecium possesses virulence vectors, including the surface proteins and polysaccharides, contributing to its pathogenicity. Enterococcus faecium can be transmitted through various ways. First one, person to person, and second, contaminated surfaces, and third, contaminated medical devices, and fourth, contaminated healthcare environments. Enterococcus faecium can adhere to the invade various host tissues. Adherence is the initial step where the bacteria attach to the host surface, often facilitated by the surface protein, and the invasion involves the penetration of the host cells or tissue by the bacteria. Enterococcus faecium can contribute to tissue damage through several mechanisms. First, biofilm formation, toxin production, inflammatory response, and intracellular survival. Enterococcus faecium is not typically associated with the direct formation of ulcers. It can contribute to ulcer formation indirectly by causing infections that lead to tissue damage and inflammation. Enterococcus faecium is not commonly associated with causing lymphadenopathy, which means swollen lymph nodes, while Enterococcus faecium can cause infections, especially in the gastrointestinal tract, bloodstream, and urinary tract. It typically does not have the predilection for causing significant lymph node enlargement. The incubation period of Enterococcus faecium is 24 to 48 hours, and the clinical disease caused by the Enterococcus faecium can cause various clinical disease, especially individual with compromised immune system or underlying health condition. It is often associated with healthcare associated infection, including urinary tract infection, bloodstream infection, and surgical site infection. And Enterococcus faecium is known for its resistance to antibiotics, which can complicate treatment and make infectious more challenging to manage. And laboratory diagnosis of the Enterococcus faecium typically involves obtaining samples from the affected sites. The specimens we are collected from the test of Enterococcus faecium is blood and urine or wood cultures. Key diagnostic methods are include. First one is culture and gram staining. Culture, it means isolating the bacteria from clinical sample on specific agar media. And gram staining, identify the bacteria gram positive or gram negative about the characteristics. Biochemical test, confirming the species through the various biochemical reaction. And antimicrobial susceptibility testing, determining the antibiotic resistant profile, as Enterococcus faecium is often resistant to the multiple antibiotics. Molecular diagnosis, PCR or other molecular method may be used for more accurate resistant genes. Vancomycin and tactomycin is used to treat the Enterococcus faecium infection. Thank you guys for your patient listening. Next session we are discuss about another bacteria characteristics. Keep support my channel. Thank you.